till today how to develop a line or how to develop a variety through various approaches. We have discussed mutation breeding, we have discussed advanced genome editing, we have discussed molecular breeding methods to develop a variety, and we have discussed all conventional methods to develop a variety. We have discussed hybrid breeding, by utilization of hybrid breeding, how we can develop a variety. And finally, we have a product. But how do we proceed with that product? You want to show to the world, okay, you have developed some product, and you should convince the society, or you have to convince the farmer, okay, my variety is best, and you should adopt it, and you should replace the conventional variety. Now, how do you will do that? Normally, for example, see how many breeders are working in rice itself. I think last year, last February, one conference was happened. I think February rice workshop. You know, for rice crop only, nearly 200 and plus scientists came. Only rice breeders across the country. They have assembled, and they have decided some variety has to release. Like it's a workshop, ASAP workshop. See, like when you take nearly thousands of breeders were working in the public sector only. Think about private sector. Think about any NGOs or CJR systems. See how many number of breeders are working, and they're all working in the same crops, like rice, major cereal crops, and some pulse crop like rice, maize, wheat, and how many varieties are developing. Okay, but farmers are using only few varieties. Few varieties. But like everyone will release the variety without any system or without any pipeline, without any legal things. And what will be the situation of farmer? Like how we can choose the variety, how we can choose the best variety, and this is the things will be a very complex in nature. And also that's why Indian government actually made some rules and regulations, and how we can release a variety, particular developed variety, and how we can bring that to into breeding chain. We will going to discuss today. That is called as system of varietal release and notification. Then before 1966, actually varietal development was either belongs to state, either an individual can do, organization, scientist based. It's like informal affair. Okay, you developed a variety, you multiply it and you distribute to your known farmers. Then if it is a best variety, it will go on increasing, and the area will going to increase. It's just a normal procedure. But after in 1966, like when the farmer started to adopt a very good seed, and the seed replacement rate was slowly increasing, like 1966, we know it's the era of green revolution. At that time, this the legality, like this system came to know, actually, like government has passed bill to make this in a streamlined way of releasing, like it should be a legal affair and should be a not informal affair, and the farmer has to get the best seed only. Then in 1964 government thought that they actually formed two committees, like CVRC and SVRC. If the varieties has to be released in a national wide, then you have to take the approval of CVRC, and you have to follow the procedures which laid by the CVRC system, which means Central Variety Release Committee. Then, if you do want to release a variety for a state, particular to some state, you should go the guidelines along with the SVRC, like State Variety Release Committee. And in 1968, some function of this CVRCs and SVRCs actually transferred to be one seed committee. Okay, in 1966, which act was passed?
Seed Act, sir. National Seed Act.
Yeah, Seed Act. Okay. Then, which clause is actually important for the notification of variety? Which section of the Seed Act? How many sections are there in Seed Act? So you should remember all those things, 25 sections. And which actually section 3 will deals with the central seeds committee. Section 5 is a notification of variety. Okay. Section which is seed inspectors. Okay. What is roguing, where it will come. Laboratories based on which section has this been set up. All those things should be in the tip, should be in the fingertips. Okay. Then only you can crack the exam. Understanding and knowing the concept is important. Along with that, you should aware of all the things, and you should some things you need to keep in mind before going to exam. Like that is also very important. Okay, along with the understanding, you should also remember some things. Okay? Even though it looks simple, it's very important. Then, then when you look the overall uh, the the pipeline, how the overall system, how in India the varieties are released and which methodology you have to follow. In nutshell, we will discuss. Like first, any either ICAR institute or any non-ICAR institute or seed industries or public-private partnership, whatever it may be, if they are developing a lines, either it by farmers, individual plant breeder, whatever it may be, they had to go with coordinated trials, All India Coordinated Crop Improvement Project. Like when it is started,
1957.
1957. Okay, which varieties were first released through ASRPs?
Initially maize, sir.
In year
61, I think, sir.
check it, okay. 1966, I think. Once you check with the Ford Foundation, like we have done this. Okay, then, first, after like in All India Coordinated Pro, you have to submit application. Then they will first you have to do regional trials. Actually, in your stational trial, you have to do after confirming stational trial, you have to approach ASARP. Then they will conduct initial varietal trials, then advanced varietal trial one. Then they proceed to advanced varietal trial 2, then again it will go for varietal identification committee, then it is evaluated further by central subcommittee on crop standards and notification and release of varieties. Actually it is constituted, this body is constituted by central seed committee, and you should know which section have a provision to formation of central seed committee in the seed act, you should know. Then section 3, I think. Then after that it will go for release and notification. We will discuss one by one. First, we have developed variety. You should, when you are really you want to release your variety for a whole nationwide, what you have to do? First, you have to uh, supply the nuclear seed, or you have to supply whatever the seed you have developed to their stational trials. Then after stational trial, which are the lines you are promising, like showing superior performance, they are entered into the All India Coordinated Trials. What is All India Coordinated Trials? ASARP and RISE situated at headquarters is situated at
Hyderabad,
Hyderabad IIR. ASARP and groundnut.
Junagadh, Gujarat.
Like this, you should know for each and every crop. Okay, it's very very important. Okay, and it's mentioned in the all plant breeding books. At the last, in appendix, in Pundan Singh, BD Singh, whatever the book you take, this information is mentioned because this is very important to know. Okay, then because why you are under the system of ICAR, you should know which are the ICAR institutes are there across the country, and which are the which system you are following for varietal release, wherever the crops and where ASRPs were headquarters are located, and which are the recent varieties are released in each and every crop, that is must for ASRP exam. Then after in ASRP trials, how the trials will be conducted, it should be like initial varietal trial. In that, it is a first year initial varietal trial where actually, like I am telling continuously, you have to evaluate the variety. How do we evaluate the variety? You should have some benchmark. Na? For example, you are comparing with someone only. Okay, you want to evaluate some person, we should either you have to say it is intelligent or not. How you will check? You should keep some benchmark as a mark. Then based on that, you have to evaluate. Like the same way, when you are evaluating the particular things, you should include the checks. Like in experiments, how we will include the checks. In the same way, you should include the checks. And deciding the check itself is a very difficult task in most of the like ASRP trial. Actually, they will quarrel. Actually, like when you attend the workshop, I think if you attended the workshop of ASRP, you will come to know. Even breeders will quarrel like anything. Like for their the data is like they'll raise the many queries. Actually, it will go for hours of discussion for deciding only the check. Means, for example, you want to release particular variety in a multiple states, then you should include which check?
National check.
National check.
National check. Then again questions come. Which is the national check? There are so many mega varieties are there. Then which can be taken as a national check? For example, some varieties are developed for higher protein content, but not that much as promising for the yield. Then for that you have to take which check. There are so many discussions and so many. It's looks simple. Actually, developing a high varieties is like we have standard methods, but to deciding these things and league proceedings is very difficult task. And deciding national check, if you are going if you want to release in a national wide, have to decide the national check. If you want to particular zone, for example, you are developing a salt tolerant variety of rice, either you can release that one variety to all over the India, but you're developing a salt tolerance mechanism, but yield may not good as other variety. Like some varieties are very highly responsive for high environment, but you are releasing a variety for salt tolerance. Means you have to release the particular variety, salt tolerant variety, where the soil is affected with salinity. Then you have to see which zone is affected with the salinity. Then you have to identify which is best performing variety in those area. Then you have to compare with that variety. Then you should take the best performing in that region as a zonal check. Understood how you select the zonal check?
Yes,
First, you
sir.
have to identify the zone. Then, in that zone, which is the best performing variety till that time for that particular salinity condition, that variety you should use as a check. Then we will test. Then your variety should be superior than those checks. Okay. At least we consider it is not benchmark. You just take approximately. At least it should be 15 to 20% higher economic yield than the which check you are using. Okay. Then, if you are want to specifically for specific area, you can use local check. For only some areas of you want to use. Then, at least minimum three testing locations for each crop is very much required. Means like at least they have to go with multi-location trials. Then, after that, everything like it should be evaluated by the team for agronomists, pathologists, entomologists, breeders, and the project directors. Okay. Then after that, if the variety is best performing than the check, it will advance it for the another yield trials. Okay, advanced varietal trials. In advanced varietal trials, normally generally like 16 entries per check will go along with the checks. Like we will conduct a test in a CRBD designs. Then with minimum 3 to 4 replications are used, 3 to 4 locations we need to use. Then same way, like you have to select the best individuals performing better than the check. Then you are going for another AVT2, third year of testing. And many times it asks at least minimum 3 years of testing for each variety is must for varietal release. That you should remember. One is initial varietal trial, another one is AVT1, and another one is AVT2. Then there all the quality data or all the better yield data, everything is taken. Then you have to go for release. uh, you have to propose that variety. Okay, this variety is in both these IVTs, AVT1, AVT2 is performing superior. Okay, this variety may be regarded for the release. It is just proposal. Okay, it is not decided. Okay, it has to be released. Then this proposal went uh, go to the your committees. Then that committee has make one team. with consisting of several members. Then this committee will decide, okay, your variety has to be released or not. Then they will submit that to government. Then government will give gazette notification. Then how this process will, like after 3 years testing, then which is the best variety, there go and and breeder has to supply sufficient seed for large scale trials, like seed kit trials. Normally, like seed kit trial will follow in the farmer field in a more than 10-hectare area, evaluation again done. Then they will decide, okay, this variety for, they will check for value for cultivation and use. Either this particular variety is agronomically uh, good, or yield, in terms of yield it is good, and means one term you should remember, value for cultivation and use. After deciding, okay, this variety is having value for cultivation and use, then it is go for identification proposal. Then actually breeder has to submit 15 days before annual workshop. Like they had to send the application to workshop 15 days before only. Then they will do all those that they will analyze these data. Then they will recommend for release. It's a very complex thing, but you should understand. Then if for the same, for example, if it is for national, in state system also, in this within the state you have to do trials. Here national wide trials were conducted with the national check, journal checks, and all these checks. But here in SAUs or KVKs, inside the state only, it has to be all first year trial, second year trial, third year trial conducted. Then there will be state varietal evaluation committee will be there. Then they will submit this recommend variety for central states committee for release. Then the, they they will decide. Department of Agriculture of State Agriculture University, they will decide, and they will start distributing the seed. But one important thing you should remember, only Central Seed Committee can notify and release the variety. Here it may directly release for seed multiplication. But all seeds which are in the seed multiplication chain, it is not notified. It should be notified only under Section 5 of Seed Act in by Central Seed Committee only. That you should remember. Then how do we notify the variety? After proposal submitted, then again based on 3 years data, they will check again, they will propose for the government through gazette notification, okay, this variety is released for particular area or a particular state. I think if you see anyone saw the gazette notification of any crop release, you will get to know. Please search, like in Google, you just search gazette notification of crop release or varietal release. In many gazette notification will come. You just observe the document, how they will release the variety, which is the head for releasing the variety, the register, the DDG of crop science, like everything in every information will be there in that gazette notification. You just observe the thing, you'll get to know many things. Okay. Then after that this like DDG crop science along with this 17 members team will decide, okay, your variety has to be go for multiplication or not. And actually there is a, I told, like, as I told, value for cultivation and use, this criteria should be meet. Every variety has to meet this value and cultivation for use criteria. Then only after it will go for notification via gazette notification with the government of India. And this all done in the provision of Seed Act 1966. Then after that, it will include it into the seed chain and breeder seed production. Like first it will nucleus seed, then breeder seed, foundation seed, and certified seed. Then finally it will reach the farmers. This is how the entire system of varietal release actually laid by the government of India by along with the ICR for the streamlining the varietal release in India. And this enables the only best variety will reach the farmers, and farmers are benefited from this pipe, actually this stream. Okay. Then when we summarize, first it development, then it will go for initial varietal trial, then advanced varietal trial, then advanced varietal trial 2, then varietal identification. Then there will be committee will set up, proposal will send, then state seed committee actually organizes one committee. They will decide the either the variety is meeting the value for cultivation criteria, then it will go for release and notification. Then it will enter into the seed multiplication chain. This is how the varietal release system takes place in India. Understood? Any doubts from this one?
Hmm.
Understood? or not? It's simple.
Yes, understood.
Your voice is breaking. Hello? Hello?
Understood, sir.
No, you just type it. Your voice is breaking. The many members are joined, but like we'll have some
Yes, sir.
question paper.
Understood, sir.
Yeah. Now, okay. Then we will have some question paper discussion.
Sir,
Yeah.
sir, if you want to release in state, so we'll go for IVT, AVT1, AVT2. Then we
But
will
within
propose
the
for,
state, within the
yes,
state
sir.
only you have to. Yeah.
Sir, then we will propose for then ASRP
Yeah.
or
No.
then,
We will not go for ASRP. It will be within the state only. Okay.
sir, then we have to
State
submit
is
3 years data.
yeah. 3 years data you have to submit.
Sir, then,
State but notification we can't do. If you want to
sir,
notify that variety, we should go for CVRC only. Okay? So
sir,
you have to
sir, but,
approach. State.
sir, but that 3 years data can't be manipulated.
No. Yeah, actually, it is the biggest challenge. Actually. See, no. Here also KVKs and SAUs. See, for example, in Karnataka state, how many KVKs were there? How many SAUs were there? Everywhere they will do the ASR like these trials. Okay, within that, normally because now when breeder will submit the varietal name, never he will going to release. There will be numbers they will give. Okay, there is and randomization and there is a one team always while because to avoid this bias they will constitute a team of breeder those the breeder developed that variety along with other committee they have one team then everyone will come and check either they are maintaining properly either all the field condition correctly or not either they are following the experimental design or not either they are following randomization or not everyone actually they will take care much actually because now yeah as you tell for example I don't for example you are also breeder and I am the evaluator in my center if I don't like you means definitely what I will do I will make you that your variety is not like it is not I will manipulate the data in such a way that you are not your variety is not performing good that bias will happen okay that to avoid that normally while giving these entries will give entry number like even I have attended this workshop actually it happened in New Delhi only like in our campus only the KSRP and rice everything like when they're presenting the slides it's everything is number you will not get anything every breeder will come now actually they are having their numbers now whenever their slide will come where then they will note it down what is the yield and all the things will be there. They will just note it down. And when was the trial? Actually, the breeder also noted down all those yield data. They will compare. Okay, in that in the report it is like this. Why you are presenting like this? They will question and they will quarrel like anything. In the workshop, everything will happen, and there are always misunderstanding. Everything will happen, but naturally we assume that it will not happen because we're
Okay,
sending
sir.
that in the numbers we're not sending any names we're just sending in the numbers and design should be maintained very properly and it is evaluated by everyone it may not happen but until can't predict
Sir, what is
Okay.
the exact difference between release and notify variety, sir?
see these means for example um you just see many varieties which are released but it is not notified notification means the under section seed for like under fifth section of seed act it should be gazette notification. I will release from the state. They will okay. They will announce that this particular variety is released for this particular state. Means seeds will be available by the particular concerned authority. I will give that gazette notification so that it will be
No, sir, I'm asking if the variety is not notified, then it is not allowed for multiplication across the nation, sir.
No, no, no. In the government system, we can't be maintained, no, released. There will be a lot of questions will come. But we can manage. Like there are truthfully labeled varieties will come. Maybe there they may not notify actually, but you should notify. Okay? But for all public sector varieties, they definitely go for notification of the variety. But registration through PPFR is voluntary, I think.
That is protection, sir. Intellectual
Yeah, that
property.
protection is voluntary. Either you can go for protection or you can not go for protection. But notification it is uh, actually notification is not that much compulsory. It has to pass through CDAC. If you want to release throughout the nation, it has to be recommended by this committee only. But otherwise, it will not be recommended for release actually. This has to be followed. But for protection, it's a voluntary. Certification also is voluntary. Either you want to you want to maintain the certification standards, that's up to you. But I think notification is compulsory. I will check once. But certification and protection is not compulsory. Wait, is it notification? We'll have some questions today. The screen is visible?
Bulk breeding method is visible, sir.
Okay, okay, okay. Otherwise, actually, paper was missing. We will download otherwise. We will deal with the QTLC. Yeah, any doubts from sequencing? No doubts. Huh?
No, sir.
We'll have some. What is maintenance breeding? I'm searching slide actually. I'm missing this. What is maintenance breeding? No, no,
Like,
no.
sir, we will maintain inbred lines like that. We have to
Can't maintain
maintain the
variety.
purity of the variety or the inbred line.
Can't
Purity of the
maintain
variety.
variety. Only we can
Inbred
breeding
line
lines we can
variety,
maintain.
anything, sir.
Everything, sir. Hybrid
Everything.
varieties.
Okay, when you are
We
defining,
need to maintain purity of
yeah.
everything.
Okay, when you're defining, you should be very perfect. When you say only maintaining inbreds, then the question comes: all varieties which are released cannot be maintained through maintenance breeding. That question will come. Should be answered very properly. Yeah. We'll discuss some of the provisions of IPRs. Then tomorrow we will discuss in detail.
Sir, for PGR, what material we should cover, sir?
Usha Kumari. Usha
Can you
Kumari
repeat,
book you
sir?
have to read. Huh?
Usha?
Usha Kumari. Plant Genetic
Okay.
Resource by Usha Kumari. You not followed my SRF preparation strategy video. There I have mentioned in
Yes.
detail about which book we need to refer. That is very, very important. Usha Kumari book and all those books are very, very important actually. Yeah, now my slides are visible.
Can you share the books or PDF?
Actually, I don't have that book PDF. I will share that book front page. You can order in Pusa Book House. Okay, it is very important for any ASRB exam or future ARS exams. Please buy it. Either you can print. I don't have PDF that one. Then we'll go to another topic of PGR. Like we have discussed about how varieties are released, and we'll jump into the PGR. And in this week, I'm entirely dealing with the PGR. What is plant genetic resource? What is plant genetic resource? Can anyone define?
The
Okay, leave it. Okay, plant genetic resource, you leave it. We'll talk about some other concept in the plant breeding or in the botany. There is one concept called as evolution. What is evolution?
the organisms constantly evolves. It means develop into new organisms, new species.
New species.
Adaptation of organism according
Adaptations.
to
Yeah. Whatever the definition you said is rightly. Why it is so important? Why the breeder has to need to study the evolution? Actually, when I was discussing these things, one student was actually asked in last time, like why you are discussing all those evolutionary things? It's not required for us. And unfortunately or fortunately, most of the questions in PGR came from these concepts: evolution, speciation, gene pool. Okay. I think you just check the question paper. At least 5 to 10 questions are from this section: speciation concepts and reproductive mechanism concepts, like how evolutions has been taken based on the reproductive isolation mechanism. What are the different types of speciation? You know all those things. Okay. Can you define again what is evolution?
The organisms constantly evolve into new forms or new species to adapt to the new environment
Okay. You told,
due to natural selection.
okay, means a single line or a single species will evolve to different species.
Sir, slide is still there in bulk breeding method, sir.
Hey, sorry. Yeah. No, no,
Yes,
it's
sir.
visible. Lecture one. Yeah.
Now only it's visible.
Then what happens? You told like there will be evolution of new species during the course of evolution. Like there will be gradual change occurs in an organism through any means the species develop into a new species. What are the different mechanisms we expect to form this new species? Any mechanisms?
Yes, sir. A sudden mutation may arise, and that mutation can adapt to that particular location, sir.
Yeah, mutations. This one. And any other thing?
hybridizations,
What?
others, hybridizations with other species or something like
That may be. Can you define in a very well? Reproductive isolation. And mechanism of this reproductive isolation is very, very important. Then, any physical barriers. What are the different? What is speciation? Can anyone remember what is speciation?
Sir, a subgroup of an population converting into an separate species.
That is founder effect. Yeah, this is the one form of speciation. What is speciation? That is one form of speciation. How many types of speciation present? Anyone tell? How many of you are referring the book Principles of Genetics book by Nestor and Simmons? Not studies
sir.
evolutionary genetics chapters.
didn't study
Population genetics chapters we read, sir.
Yeah, that also. And at last these concepts were there. Should remember, last many questions came. That's why I'm discussing. I'm stressing more upon it. Last time when you open the question paper, there are a lot of questions were asked based on the species concept, BGR. It's very, very important to know. Okay? Please read about it. What are the different speciation? I will discuss. Okay? Without wasting time, we'll jump into the topic. What is evolution? As we have defined, evolution is the process by which the population of organism change over the time. And the forces of evolution are the mechanism that drives changes for whether it may be anything. Okay. Like what are the forces can you expect in the population genetics? Or how do you work? Selection, migration, genetic drift, mutation, all the things were actually the drivers of evolution only. Okay, what it will make, all these things, what it will make in the population,
there will be changes.
Change in gene and
what
genotype frequency.
change in the gene and gene,
Genotype
one word you are missing,
frequencies.
frequency, that should come. That should, the word should come actually. These actually either it may be the mutation or it may be the migration or it may be genetic drift. Okay. This should be what it will make, it alter the gene and allelic frequencies of the particular population so that a particular allele of the population is fixed and another allele either it fixed or eliminated. Means your mixed population divided into distinctive two population. That is called as your evolution. Okay, these are the driving process of evolution. Okay? The change in the frequency of the gene due to genetically, when you're talking genetically, this should be the underlying mechanism of evolution. If some species is going in developing into two different species, then definitely there will be changes in the genetics of the particular organism. That changes has been brought in the population of the organism by these mechanisms, either migration, mutation, genetic drift, or another important is your selection, natural selection. Okay? That you should remember. Then it's a and evolution is not a speed process. Like within a day or within a week, it will not happen. It is a very gradual in nature, and it's a very complex. Either you can't say that it's only through genetics. No, it's only through environment. No, it's an interaction of the genetics and environment. Then, like sometimes it's not a static process. For example, evolution has happened now, it stopped now, next nothing will happen. It's not like that. Or evolution, it having some destination, up to that it will take place, after that it will not happen. It is not like that. It's a continuous process. Should remember, evolution is a gradual and very dynamic or continuous process, and it is very complex mechanism that we should remember. Then last year in AS survey, the 10 marks the question was asked about this topic. And you should take the example of the brassica. How brassica has been evolved into different species through this mechanism, evolutionary mechanism. That like it will comes under the domestication. The domestication is a one form of evolution only. Then the main drivers of these mechanisms were mutation, migration, genetic drift, or natural selection. Then how evolution occurs? Because there is a change in the gene pool or the collection of the gene, and the gene frequency will change because of these forces. That's why your evolution will occur. Then one important thing we should take care, genetic drift. It also called as evolved rate effect. What is genetic drift? Can anybody define when the genetic drift will happen?
Due to any calamity or some environmental condition, some person is lost, then it will alter the phenotype or gene frequencies in that relation.
When the population is large, the genetic drift can happen.
No, sir.
No,
Sir, when small population is present, then the genetic drift will happen.
Yeah, that should be come. For example, why the people are so much worrying about land conservation? These things are not much important in nowadays plant breeding, but these things are very, very, very important, and everybody study about these things in conservation of wildlife. You know, why the people are worrying about lion conservation? How much the population of lion? It's hardly 1,000, at like 900, 800, below 1,000. Means there will be a lot of inbreeding will takes place. It will become more susceptible for some disease will come. Definitely entire population will going to die. Means in future you are not going to have any lions in the country. If um, any disease will occur, because if it is susceptible, because continuous inbreeding, it will happen. That's why you need to maintain the genetic diversity within a small population. Maybe some drift, for example, as you told, some calamities happen. Already there is a small population that too located in only some location of the area, like Gir forest. All the most of the lions are concentrated there. Some forest fire happen in the Gir forest. What will happen? Then definitely some lions will survive only one or two, and they will give the next population. Definitely there will be the genes which are present in that lion only will form the next population. Then definitely what will happen? The shift in the frequency of the gene throughout the entire population to another frequency of the population from the only single population. That also we'll discuss. That is called as bottleneck neck effect also. For example, what will happen? We'll discuss that. See, for example, when you have a there are two effects will come. For example, one effect is founder effect, another one is bottleneck effect. Founder effect means some particular species or a particular individuals, they will form a new population. See, in the original population, how many colors were there? So many colors. But this yellow and green, they may not breed, they may not share the gene with any other of these individuals. They only intermate among themselves. Slowly during the time, this developed into distinct population. All over this blue color, brown color, everything gone. This is called as founder effect, and as I told, what is genetic drift? Whenever the population is small, any changes or any environmental changes occurs, the complete vanish of this frequency. But if any someone will remain, like this, any someone will remain, and then definitely they will go and develop the population. For example, you take here, the only red is coming in the bowl. Then you can see here equal frequency of both yellow and this one, orange. Then when the population is very narrow, see the bottleneck, that's what is called as bottleneck. See, in the bottle inside, it's a very large population. But when there's small population, it's like a small neck. At that time, anything will happen. Only surviving individual give rise to for the next production, like next population. See here only which color is dominating?
Red color.
Red is dominant.
Red or orange color is only dominating. This is called as genetic drift. Here, the change in the gene frequency, and that you should define in a very clear way. Here, not in a single direction. Means you are having some direction which is not following. It's a non-directional change. See, at what direction the particular frequency of the allele will going to change, that is uncontrollable. That's why when you are defining genetic drift, two words should be very, very important. It's a non-directional, and also when the population is very small. This should be two words very important. When population is small, due to anything happens, there will be non-directional change occurs in the population. That is called as your genetic drift. That situation happens when this small population, like bottleneck condition. See here, very small frequency of the individual will be there. Something will happen. Then definitely there will be non-directional change happens in the population. See here, resultant population is different from this population. So only red is dominating, but earlier all the things are equal proportion. This is what the founder effect and the bottleneck effect, and how this will bring the evolution we understood. Like it will bring the changes in the gene frequency. Then this particular species evolved into different individuals. Okay, understood these effects?
Yes, sir.
Yeah. Then next comes to natural selection. And we have discussed in detail about what is selection, what is types of selections, artificial and natural selection. We will discuss little about and these natural different types of natural selection. And that class I have skipped this. And today, I think today we will discuss this one. How many slides are there? So this question was asked in the for 10 marks in the SRB exam like this year, last year, 2023. Then what is selection? Can you define?
Differential rate of reproductions.
Difference in the rate of
Reproduction of individuals.
reproduction of individual.
Individuals.
That's when you are defining, you should define in a correct way. What happened? What does that means?
That means each
Only
individual
some
reproduce
individuals.
in different rate.
Yeah.
Only
For
some
example, this
individuals.
individual, survival of the fittest. That you can take in natural selection. This having capability of more survival or more adaptable to the changing climate or whatever it may be, then it will going to survive in the next generation. Rest are other selected against. It is selected in favor, but others selected in against. Means this differential rate of reproduction, this individual only can able to reproduce and can pass this gene to the next generation. But rest other are not able to do reproduction and they are not going to share any gene for the next generation. This is called as selection. Then Either it may be through natural selection or through artificial selection. In natural selection, there are different types of selection is there. Directional selection, stabilization selection, and disruptive selection. Then, what is directional selection?
Sir, if either dominant or recessive phenotype
Either
is
of
selected
the
directly.
genotype is selected, sir. Either of the extreme genotype is selected.
Extreme, yes, sir, extreme.
See, when you look into this graph, for example, when you want to just check this one, actually, this one, giraffe, the long necked, here the direction of selection towards long necked giraffes. Means only those giraffes having long neck, they are only surviving, and this is called as directional selection. Means your selection is directed towards the any one of the phenotype. That is called as directional selection. See, here in the lizard you take, only long tail lizards were favorable, selected in favor. Then at the end, during the evolution, only long tail lizards only present. Okay. Either any extremes are selected. Only one of the extremes are selected. Then it is called as directional selection. Means you just check the graph, your mean is shifting. Your mean is shifting towards the one side of the direction. For example, long tail. This is called as your directional selection. There is another form of selection is called as stabilizing selection. See, here the mean is shifting towards the stable form, average individual. See, the frequency was increasing, those individuals performing average. Here it is increasing like this. Only the average individuals are selected in favor, their frequency only increasing. And that graph is very clear, you can observe. See, for example, here both the extremes are selected against, only those who is having the normal medium size, their frequency will going to increase and they only evolve. It's a best example can anyone give in humans for stabilizing selection. In many paper it discussed. For example, in a mother womb, what will happen when after delivery? Normally, what will happen? If the child is very, very less weight and overweight, they will not going to survive. Those having medium weight, they will only survive. Okay? This is only stabilizing selection. Understood what is stabilizing selection?
Yes, sir.
Then another form of selection is disruptive selection. Can you look into the graph? Here, average individuals are selected against. See, there is no increase in the frequency. But these extremes, either it may be short, either it may be long, their frequencies are increasing. Their population frequency is increasing. Okay. Then this is called as when both the extremes of the population increases. Means the selection is in the direction of both the extremes, then it is called as disruptive selection. Understood?
Yes, sir.
For example, when you have a short tail, their frequency will increase, and only long tail, their frequency will increase. Those who having medium tail rats, they will disappear in the future. This is what a disruptive selection takes. Understood what is the types of selection? Types of natural selection? Directional,
Yes, sir.
stabilizing, disruptive. And what are the mechanism of evolutions, driving force of evolution, mutation, gene integration or migration, or genetic drift, natural selection. And you should remember what is genetic drift, and you should remember what is founder effect, what is bottleneck effect. Then other forces of evolution, polyploidization, co-evolution, selection pressure, or epigenetics. We will discuss in detail about these things tomorrow's class. Okay, any doubts?
Sir,
Up to
another
here?
type of selection is there, metacyclic selection. What is that, sir?
Yeah, that will there. That is there. Okay. The cyclic selection is also there. There is a number of types of, but majorly how the evolution will happen through natural selection by these mechanisms I explained. Okay. There are a number of types are there, but just they are the major types of selection who drives this evolution. Okay. There are a number of types are there. In different books, there are different classifications, but major these things. Okay. Then tomorrow we will discuss how polyploidization is very, very important actually. Neofractalization, genome reduction, how it happen, how it drives the selection, we will discuss. This one I just had not added this slide. That's why I'm not discussing. What is coevolution? Anybody heard about Redfin hypothesis? Co-evolution is the perfect example of one thing in the plant body. Can anybody tell?
Sir, race-specific pathogen, sir, along with
Exactly,
that
exactly.
thing
Okay.
co-evolves
The
each other.
exactly. When the both host and the pathogen are when some individuals they are in association, either it may be whichever the association, mutualistic, uh, antagonistic, whatever it may be, parasitism, mutualism, whatever it may be, when they evolve together. Okay, for example, the disease uh, resistant genes and virulent genes. Best example of coevolution. Virulent genes and resistant
And
virulent genes in the host, resistant gene, sorry,
and
virulent gene in the pathogen, host resistant gene in the host. Best example for coevolution. That you should remember. Then selection pressure, we will discuss what is selection pressure. And selection pressure also typical example of your virus like a pest and
Pest.
host interaction. Selection pressure, then epigenetics. Okay. Then these things we will discuss. Okay. Polyploidization and domestication, how this will happen. Okay. This is actually this question was asked 10 marks. How actually the different types of brassica evolved due to evolution. Actually, this is human-made evolution. Actually, earlier this one only was there. Nowadays, we evolved the brassica into these many types. How it exactly happens? Okay, this asked in the exam. Okay, that's a very important. Then we will discuss next tomorrow's class. Any doubts up to here?
no, sir.
Okay. Right. Any questions apart from other topics than this?
No,
Yeah, okay. Bye bye. I think 4 or 5 classes like I will take only PGR. Then we will shift to other things, cytogenetics and quantity genetics. And basic genetics, actually, can I make recordings? Basic genetics, or can I take live class? We can attend live class also, but I may take oftenly like in afternoon or morning. Like I am thinking to record genetics classes.
Live is better,
Fine
I
time.
think, sir. Interaction.
Yeah, but nobody will come. That is the problem. Only you and Venkat is coming daily. And today at least I have so many individual, like so many persons came. It's nice to see. Okay. More, see, we have discussed very, very important topics. RNA-seq. Expected lot of questions on RNA-seq and expected lot of discussion on sequencing. Okay. It's very interesting and had to know. Okay, but nobody was there, and I just keep like I just gone through whatever the slides were there. I just finished. Whenever more number of people will get, there will be more questions and more discussion. Then we'll learn more. Just collecting information also like. Say I can't keep collect everything at a time. If a question will arise, then only I can synthesize the answer. Like I will think about that, then I can discuss. Like collecting myself entire information, it's very very difficult for me. Also. Whatever there in the books, I will just keep and I will discuss. If nobody will come, if anybody come, they will. If we discuss, then only we can expect more information from my side. Okay. Then okay, I will take live class only.
Sir, sir, when quantitative genetics might start, sir?
Quantitative genetics I will start after cytogenetics. Okay. Only genetics and cytogenetics is left over part. After that, I will take quantitative genetics. Okay.
Okay, sir.
Quantitative genetics, even I had to make very interactive slides and I had to make all the notes, okay, running notes. Okay, I had to revise all my notes. It's very difficult to teach quantitative genetics. Needs very lot of practice. Okay. It's not like that coming and telling the class. You know how much effort we need to make. See the slides you can like information is available many, like you can we have a lot of information nowadays, but arranging and which information has to be taught and like making things simpler is more difficult than keeping everything. Okay, then telling only particular topics and particular points, it's very difficult to make the notes. It's all my running notes I just converted into slides. See the slides, you can see all their points, bullet points. They're all my running notes actually. I just converted them into slides. That's why it is not like a bookish information. Like how you will write my running notes in the class. It's all my running notes of classes. So I used to make in bullet forms and only specified top points will be there. And you should always refer books and you should make your own running notes. Then definitely you will be in a very good like you'll be understanding of the topic will be very high when you are reading the books and you are making own notes and you again revise and convey it to other. It will be very good method of reading. Okay. Many people I saw just simply read and again they will revise the book. It's not the way. You should make your own running notes, either listening to class or reading the book. You should make at least for one topic. See, if for example, topic I made a very nice notes. See this one. In the when you're reading the book, how much information was there? Like these are my class running notes. See this. There are so many models that there I've taken this model because this only taught in our class. And maybe that will be very important. See this bullet points. How this will come actually? Three. When you read the book, see how many pages are there in the association mapping. Only highlighting these things, it's all my running notes of the class. Okay? And directly I make them into for presentation. That's why it look a very in a very bullet points and it summarized notes. It looks like summarized notes. So you prepare your own notes by reading the books. Don't just simply read and throw the books. Okay, make your own notes and revise many times and cover as much as objectives of every book. Either it may be Pundan Singh or it may be uh, object of BD Singh and BK Prasad, whatever it may be. Cover at least 100 to 200 MCQs every day of each topic. Then revise syllabus as many times as possible. If any word you are doubting, you just type in the Google, you get some information. Okay. In this way, how to read. Okay. Okay. Bye bye. Any doubts you can ask, or we will end up. Class. No doubts?
No, sir.
Good night. Night.
Good night.
Nice.
Night.